Good evening, ladies and gentlemen. My name is Jason Swar. I'm a senior mechanical engineering student here at Grove City College, as well as a member of the Acoustic Biomechanics team. Tonight we will also be hearing from Noah Brozovic, Matthew Gore, Libby Stewart, Bryce Walker, and Connor Young from our mechanical engineering side of the project, as well as Kim Einlay, Frederick Mayville, and Aaron Hammerhuber. Aaron is at PAX currently, so his portion has been pre-recorded. Similar to how a doctor uses a stethoscope to monitor and understand the condition of the heart and the lungs solely from the sound that they make, a single student came to one of our professors here and asked if it was possible to make a stethoscope for the feet, one that's capable of understanding the condition, the neurological and physical condition of a runner solely from the sound of their footsteps. Our professor was instantly intrigued and made it into an independent research opportunity for the student, which then became a senior capstone project, which now spans 5 different departments here at Grove City College. This evening we'll tell you about our testing methods, our results, and our future plans that we derived with one common goal in mind. And that goal being to provide a clinically useful assessment of an athlete's condition from the acoustic measurements. And we'll begin by talking about the process we used for this.
Hi everyone, I'm Noah. I'm going to be going over the process of our project in total. We have broken it down into 4 steps based on timing of how we go through our project. The first 2 are what we currently practice in our lab and the work that we take home with us, and the second 2 are our developing technologies for further use in the lab in the upcoming years. We start off with our lab sessions and lab recordings. These include taking pressure recordings with these sensors that go into the soles of the feet, which will be discussed later on too, and then video signals of someone running, and the most important part is the acoustic measurements. Then we take all this data and we find everything that we can from the data that we take to validate what we find through acoustic measurements and recordings. So everything that we use other than the microphone is used to validate what we do find with the microphone. The third step in our process is the development of our load cell system, which is being used to measure the lateral forces of a runner running on the treadmill, which we have developed because there is no other method that we've seen, and we currently don't have anything in our lab to be able to do this. The last step is using MATLAB and specifically biomechanics of bodies to have a full 3D animation of a runner as they're running, and we'll be able to predict muscle joint forces and tendon forces throughout their body, throughout the whole motion for the recordings through that. So overall in the lab, a overview, our goal for this semester was to test 20 subjects in our lab. We made it to 18, a total of 20 subjects, 10 female and 8 male. We had 20 total recordings with 2 repeats due to discrepancies in data in our early test this semester. So again, we filmed their posture, recorded their foot pressure and the acoustic measurements in every recording, 8 different measurements, which we'll go over more in the next few slides. And we developed data analytics to quantify the gate changes in the acoustic measurements.
Good evening, everybody. My name is Connor Young, and I will be discussing the procedure that we follow in the lab environment. So to start off, as soon as our subject gets into the lab, we sit them down and we take a blood lactate reading. That just involves pricking their finger, taking just a little bit of their blood on a test strip, and that gives us a reading of the amount of lactate that they have in their blood right when they get into the lab. Blood lactate is just a measure of essentially how fatigued any person is at any given moment. The more lactate that you have in your blood, the more tired you are. So once we get the blood lactate, we then strap them up with a heart rate monitor so that we can monitor their heart rate throughout the lab process. And then once we get all the equipment on them, including the shoes, and get our video cameras all set up, we get them onto the treadmill and we start the lab procedure. There are 3 main parts of the lab procedure. There is the pre-fatigue round of data collection, the fatigue protocol, and then the post-fatigue round of data collection. The pre and post-fatigue round are exactly the same, the only difference being the level of fatigue in the subject at that time. So the pre-fatigue section, it consists of 4 data collection segments, each a minute long, and that entails the subject moving on the treadmill at any given speed. We start at a speed of 2.8 for a walking speed, then we record that data for a minute, then we increase the speed to 6.0 for a jogging speed, then we increase again to 7.4 for a running speed, and then we increase again to 10.0 for a sprinting speed. After we collect all that data for the pre-fatigue, we get them off the treadmill, we get all the equipment off of them, and then they get back on the treadmill for the fatigue protocol. The fatigue protocol is just what we follow in order to take our subjects to a suitable level of fatigue so that they're sufficiently tired and we can see a difference between their pre and post fatigue data. So for the fatigue protocol, we start them out at a speed of 6.7 on the treadmill, and they will run at that starter speed for 2 minutes, and at the end of that 2-minute segment, we ask them to assess their rate of perceived exertion, or RPE, using Borg scale. They will most likely start in the 7 to 9 range as a pretty light exertion. As long as they're below an RPE of 13, at the end of that 2-minute segment, we increase the speed by 0.5. We then continue that 2-minute segment assessing the RPE until they reach RPE of 13, at which point we stop increasing the speed and we maintain that speed for the remainder of the fatigue protocol. We continue to ask them to assess their RPE every 2 minutes until they reach an RPE of 17. Once they hit that 17 mark, we write down the time, and then 2 minutes from then, the fatigue protocol is over. They get a 2-minute cooldown if they want it, and then we get all the equipment back on and get them back on the treadmill for the post-fatigue testing. In between the fatigue protocol and the post-fatigue testing, we do record their blood lactate once more and get their heart rate once more. And then, as I said before, the post-fatigue is exactly the same as the pre-fatigue with those 4 speeds, 2.8, 6, 7.4, and 10.0. And then we collect all that data, get all our equipment back from them, and they are done. Now we will be moving to talk about more specifically how we collect data in the lab environment.
Good evening, I'm Fred, and there's a number of different technologies that we use in the lab to collect our data. The main one, of course, being our acoustic data, and that's collected using a microphone that's situated directly in front of the treadmill to collect the sound of the subject running. Secondly, we're also using a technology called F-scan, which consists of these films like this that go into the soles of the subject's foot, and they measure the pressure forces as the subject is running, and that's collected using that small little pack on their back, strapped to the back. And then lastly, we are also recording our subjects with 2 separate cameras. One is a GoPro camera, that wide-angle lens. It allows us to capture the full body motion of the subject. And then we have a second high frame rate 9-Nox camera, which allows us to focus in on the stride and see how the subject is running and landing on their feet step by step. For the acoustics itself, there's a number of different things that we needed to do to try and suppress any noise that there might be in the room or caused by other sources, so that we can get as much data out of the acoustics as possible. So this here is a picture of our new lab down in the basement of Rockwell, courtesy of the school. And a couple of the things that we did to it was lay foam matting on the floor and acoustic panels on the walls, just trying to lessen the reverberation of the room as much as possible, so we get we don't have as many sound bouncing around and entering the microphone at different times. And then after lab, we put all our acoustic data through a filter, and that's just to get rid of all the constant whirring and squeaking and stuff that the treadmill itself makes. and get that all out and produce clean steps where the foot is landing. And then you can also see that foam structure right in front of the treadmill. That's where our microphone is inside, and that's just to prevent, again, any echoes coming in from the back or the side that we don't want. We just want everything direct from the foot going towards the microphone.
Yeah, so when we take this data, there's a lot that goes into it. When we are validating it, we validate it by aligning the acoustic signal with the F-scan sensor. My name is Livia, I will be explaining this. So as you can see here in this image, we have aligned the acoustic signal with data we get from the F-scan. We had a junior student working with us in research, and he helped contribute to kind of this advancement in the technology and in the analytics. So you can see here the green signal is the filtered acoustics, it's the full acoustic signal, and then the brown outline is the envelope. So basically what an envelope is it is a reduction of the number of data points while still maintaining the shape of the acoustic signal. So we can align the acoustic signal with the center of force velocities from the F-scan data to kind of try to understand what is happening. So this alignment, you can see the red triangles are the right foot and the green triangles are the left foot. As the right foot is lifting, the green foot is planting, and we can identify this within the acoustic signal. So when we are identifying parameters from the acoustic signal, we can identify what is the left foot and what is the right foot, and then evaluate any differences with fatigue.
My name is Aaron, and I'll be discussing the effects of fatigue on a runner's gait. We've got some images right here showing pre and post fatigue of the average force on the runner's foot. This is over the jogging section, and it's over the course of 1 minute, measured with the F-scan sensors in the bottom of the runner's soles. We can see some clear differences here, starting off with this red blotch right here. In the pre-fatigue, it's rather small and located on the ball of the foot, but as we get to fatigue, we can see the runner starts to use the side of their foot more. And this is something we noticed in the lab, that this runner tends to roll the side of their foot. So this is something that we automatically picked up in the lab as being more pronounced when they're being fatigued. So that's a very interesting piece of data. They also tend to heel strike more on this left foot here. We can see that with this orange versus this green in the pre-fatigue. We can also see a lot more emphasis on the arch on the right foot as opposed to the outside. They're actually taking pressure off the outside of their foot and putting it more on the arch. So that's something that we didn't really expect coming out of the lab, but it's something we noticed in the data, and it's very intriguing. So we can see some clear differences between pre and post fatigue in the F-scan data. But what about the audio? That's where this project really lies. So let's take a look at some average audio envelopes. Envelope being the reduced downsampled signal that is more simple and easier to understand. So we've got the envelopes of a bunch of different signals here, all the signals for that same set of data. We're seeing an amplitude of around 0.05 right here, around halfway between 0 and 0.1. And then we're seeing rather uniform signals. They all seem to be about the same. We've got some small outliers up the top here, but in general, they're all around the same type of signal. However, when we get to the post-fatigue, things start to get shaken up a little bit. We're at around 0.1 amplitude, so we've got almost twice as much volume from this signal, and that's not even counting all these tons of outliers we've got here. We've got a wide variety of different signal shapes and sizes, as opposed to the fairly uniform set that is going on up in the pre-fatigue. So post-fatigue, we're seeing a lot more variance, a lot more interesting dips down here. We see the volume is starting here and then dips down and then comes back up near the end. And so that's not something that we quite see in this pre-fatigue, but it's something we do see in the post-fatigue. So this is very exciting for us, as our project is not only to find clinically useful gait parameters, but also to assess the difference between pre and post-fatigue using the acoustic signals. And these two charts pass that objective in flying colors. We can easily tell right away that these signals are way different than these ones. And so we're easily telling the difference between pre and post-fatigue, and that's super exciting for us. It shows that we really did meet our goal as a project, and it's something we're very excited about being applicable right now, not just in the future, but we can apply this technology right now, and that's something that's very exciting for us.
Hello everyone, my name is Kim, and I will talk more about part of analyzing data. So as Libby and Aaron talked about for the past 3 slides about envelope and then the importance of that, I just want to emphasize it one more time. Enveloping technique is the way to reduce the number of data points without losing the main characteristic of the signal. For example, we don't want to lose the peak of the signal, we don't want to lose the minimums of the signal because that's where we know the foot is landing or the foot is lifting. So with normal enveloping technique, for if you're recording data from second 4 to second 10, it's enveloping reduce data to 10 times. And this semester I discovered that if you do envelope and then you also try to find the peak of the using peak analysis, you can reduce the number of data points to 100 times. So I have like way less number of data points while still picking up the maximum and the minimums of the data. I was trying to find foot contact time, and foot contact time is measured by the time between your step when your left foot, for example, when your left foot lifted and your right foot landed, minus the time when neither of your feet touching the ground. So as you see on the graph on the left here, it is the time between the step. I discovered that from aligning acoustic signal with F-scan signal, the time between your step is the time between the 2 minimum points. By measuring that, I was able to plot into the right graph. The right graph is a comparison between the pre-fatigue and the post-fatigue. The pre is the blue line, and then the post is the orange line. So one thing I discovered is not when you fatigue, you're not spending less time on the treadmill, but rather it is your form. When you get tired, you get sloppy. So you're spending less time on one foot and just try to get back to the other foot just because the other foot is stronger. So as you can see here, you see 2 big dips on the blue, on the orange line, and then it just seemed like after that, the runner was able to get back to normal. One of the challenges that I encountered when trying to find foot contact time is the size of the data sets. So as Connor mentioned, that for every speed that people running, they would run for a minute. But because the data set is so big, I can only every time the algorithm run, it can only run the data from like 2 seconds to like 6 seconds. So as you see from here, I only have the data from second 4 to second 6. Yeah.
Hi everyone, my name is Matt, and I'll be talking about our last parameter, which is called sample entropy. So a brief overview of what sample entropy is. It's basically your complexity or your repeatability within a time series signal. So this was originally developed around the concept of a cardiac signal. So in a hospital, just looking for differences within that pattern as time goes on to be able to tell, is there an issue with this heartbeat or is it normal? So obviously this would apply to our project directly because we're kind of looking at the same concept. We want to see between the pre-fatigue and the post-fatigue if we can actually see a difference. And sample entropy is a direct way to do that. So in the fall, we tried to implement this and we ran into issues, again, like has been mentioned, with the size of our data sets. So originally we were looking at about 5 million data points for a 60-second data set. And it was going to take around 3 days to run the code, which really wasn't good for us. So when we came back in the spring with the work that was done with envelopes, we are now able to, we have a code that can run sample entropy in about 3 minutes as compared to the 3 days that it was going to take in the fall. So for example, here on the left, this is our data from 223. The blue color is our pre-fatigue, and then the green color is our post-fatigue. So as you can see, for every speed, post-fatigue is higher than pre-fatigue, which is saying that basically the signal that's coming from the post-fatigue is a lot more disorganized and chaotic than the pre-fatigue, which is exactly what we want to see. Through running this code through most of our data, we have about, like was mentioned, like 18 data sets. All but like 2 or 3 of them have shown this same trend, which is very promising. We really don't have a way to compare sample entropy to another parameter to verify it at this point, but it's very promising that it is giving us the results that we want to see. So as time goes on, the plan is to run sample entropy more and look for that same pattern.
Hi, good evening everyone. My name is Bryce, and today I'm going to be talking about the videos that we take in the lab, as well as the analyses that we do with these videos. So as Fred mentioned earlier, in the lab we have two cameras. We have a GoPro camera and a 9aux camera. The GoPro camera shoots the runner from a wide angle so that their full body is in view, and then the 9aux camera is what you're seeing right here and is more zoomed in on the runner's feet. So with each of these two cameras, we use them each for different analytical purposes. So more towards the beginning of the year, we were using the GoPro camera to look at the runner's forward and backward accelerations. The way in which we did this was using a software called Logger Pro. And Logger Pro has this unique point motion tracking feature in which you could track the motion of any point on the video throughout the duration of the video. So the way that this would work is we would take the GoPro videos into Logger Pro, and Logger Pro then splits the videos up frame by frame. So the GoPro shoots at about 30 frames per second. So then Logger Pro will split up that video 30 segments for each second. Then on Logger Pro, we will track the runner's foot and advance the video frame by frame, tracking their foot all along the way. So it is a very tedious process. After that is done, Logger Pro then takes this data and takes it into and creates a position versus time plot for us. So with that position versus time plot, we can then take the derivative of that plot 2 times to get the runner's instantaneous acceleration versus time for the forward and backward motion of their feet. We use the software for some other things as well, including verifying some of our other parameters, one of which is what Kim was speaking on, which was foot contact time. So to reiterate, essentially foot contact time is the amount of time that the runner's foot is in contact with the treadmill. Logger Pro makes this super simple to essentially look at how many seconds the runner's foot is in contact with the treadmill. As you can see how it is frame by frame. We can take the average of those, and then we can get a value for that and report it back to Kim so she can verify the code that she was working on. And we can do that with a couple different parameters that we were able to see visually on the videos. And then moving into the second half of the year, we got more focused using the 9aux camera, which is the one that you are seeing right here. The 9aux camera is unique in that it's specifically made for biomechanic and sports medicine research. So as Fred mentioned again earlier, it's shot with a really high frame rate at 400 frames per second, which is really useful for us because we can look at really small fragments of the video and get a good idea of what's going on here. So as you can see right here, we use the 9aux camera to align our audio and video footage. We did this in the lab. We essentially would flash record on the audio and the video, and we would clap in front of the video camera. We would use that clap to align the audio and the video, and that's the video that you're seeing right here with the acoustic signal running at the bottom. This gives us a really helpful picture of exactly what's going on with the runner's footstep at each step in the acoustic signal. We learned a couple different things from this. I think one of the main takeaways that we got from this was going into this project, we had thought that the largest peak of the acoustic signal we had originally thought was the runner's heel strike. But what we've learned with most of our runners was that the strongest peak, or essentially like the most force that they're putting down, is actually on their midfoot strike and not their heel strike. So that was one thing we learned that was pretty interesting. But overall, it's a great tool for us to see visually what's going on because we're looking at all this data, but it's easier to see with our eyes exactly what's happening here. So thank you. I'm going to pass this off to Jason.
As a few of my teammates pointed out, our F-scan system is very reliable at determining and showing us the forces normal to the foot, going straight up into the foot from the surface. However, it cannot tell us anything about the lateral forces or the side-to-side forces a runner exerts, especially as they get more fatigued. To accomplish this, our team designed and manufactured an apparatus that would cradle the treadmill on top of caster wheels. This way, all the force that would be detected, there would be no force lost to friction or the environment because it would be able to roll freely. The apparatus would also place load cells on the sides of the treadmill that are delicate enough to pick up on even the smallest force applied to them. Now, this apparatus was also designed with safety, of course, in mind, as it was placed alongside the caster wheels that the treadmill was mounted on top of are mechanical stops in case that one of the load cells that's mounted to the side became loose or failed completely. The treadmill would not actually be able to move side to side and cause danger to our subject above. Furthermore, the apparatus reinforced the treadmill by placing caster wheels along the back face and, though it's not shown in this, the front face of the treadmill. This way, in case a large front-to-back force was applied to the treadmill, the caster wheels underneath would not buckle, keeping the runner secure. Now, the data that we collect with this treadmill can be used in tandem with methods we are already using, such as our F-scan system, as we stated before. But it can also be used to validate future test collection methods, such as our IMU suit, which we will talk about shortly. However, within the last 2 weeks, about 2 weeks, we have completed the manufacturing and assembling of this apparatus as shown here, and have begun using a stand-in treadmill for proof of concept tests as well as calibration tests, so that everything is ready to go in the fall when we are able to attach it to the physical treadmill.
Okay, like Jason said, we thought we'd bring this in for show and tell. So this is our IMU suit. It's a Danish company that we got the product through, and basically IMU stands for Inertial Measurement Unit. So your cell phone has the same sort of sensors in it. It has an accelerometer, a gyroscope, and a magnometer. So those 19 sensors all feed to the control unit on the back, and then it is wirelessly streamed to the software you see in the middle there. So last semester, after experimenting with our biomechanics software, I found out that we were going to need a suit like this to be able to capture a runner's motion accurately and then predict the muscle intended forces within their entire kinetic chain. So yeah, as you can see over there in the rather embarrassing video on the left of me jumping around in a room, that motion is streamed into Rococo Studio, where you set the height and the weight of the subject and it records the motion exactly. From that point, we are able to export a file from that software into our biomechanics software seen over here on the right. And we change again the height and the mass of the subject, and then we define basically the height of the ground. And then we import that motion file. In a matter of 5 minutes, we are able to calculate all the forces going on inside of the body. So as you can see here, red means that the muscle's working a lot, and blue means it's not working that much. So testing has begun with this. We actually got it 2 weeks ago, so we haven't had a ton of time with it. But in the fall, their plan is to implement it more and really use this as a proof of concept and to validate a lot of the other parameters that we're looking at.
This project has been involved in many disciplines other than mechanical and electrical engineering. For example, we partnered with the computer science team that has created us a desktop app and an online database that we can use in the future. This is an image of the desktop app down at the bottom here. We can take individual sections of data for our walk, jog, run, and sprint. We can set up F-scan, record the data, see it in real time, and even add notes. So it seems like it'll be very important for both the audio recording, F-scan acquisition, and any other types of data we might need. We can export those files directly into the database. An example of this database page can be seen here. All the files that we've exported will be available online to anybody who's involved in the project, and you can pull them up and view them at any time. We've got the audio signal right here. This is just a piece of test data that we've got in here. And then you've got the accompanying F-scan signal in a low resolution so that it's easier to process. Up here we can select any different type of data that we'd like and step through the audio signal, step through the accompanying F-scan signal, and see anything that we want to from the data. So these are both very important tools that we think will streamline our project in the years forward and allow us to be even more efficient than we are right now at collecting data, viewing data, and processing data.
So you're probably wondering, where is this going? I had the cool opportunity this semester to be working with the entrepreneurship team that has been developing a business model plan that's going to apply the research that we've been discussing so far. So this is my team, and this is our product, the Happy Feet Foot Pod app. The Happy Feet Foot Pod and associated app would be about 1.5 inches in diameter and be able to attach to the front of your shoe via the hook in the back and tie into your shoelaces. It would then be able to listen to the sound of your footsteps as you are exercising, and just like we've been saying, take that acoustic signal and get some key parameters out of it, such as stride rate, step variance, sample entropy, and foot contact time. This will be able to assess an athlete's condition and be able to give you key information about your exercise, such as alerting you if you are running in such a way that could lead to injury. As we spoke earlier, as Aaron spoke earlier about the F-scan signals, you can see that as a runner gets fatigued, they get sloppy and they roll their feet and they do different things incorrectly. So the Happy Feet foot plot out would be taking the acoustic signal and be able to assist a runner by giving them that key information and alerting them if they are prone to injury. So these are some of our key partners during our project. We partnered with Highmark Health, Dr. Kelly Shields, and Dr. Anyi, and they were able to assist us with some of the data analytics. They helped fund our project. Also, sports medicine physician at Allegheny Health Network, and like we said before, the various cross disciplines at Grove City College. So thank you so much for your time, and we'll take any questions.